and welcome to a new episode of the Computomics podcast. With us today is one of Computomics' own, Bioinformatics Analyst Michelle Hagen.
Hi, I'm happy to be here. Hi.
As are we. Michelle, you already know we kind of like to start out of left field here. And I was wondering, what kind of creative projects do you do outside of work?
That's a nice question. Well, in my free time at home, I have several boxes of like creative stuff to do. So, for example, embroidering, crocheting, painting, drawing, jewelry making, also like ceramics, like the ones that you air dry at home. Especially in winter when it's cold outside and dark, I really like to go back to crocheting. I think that's also something that I will start this year again because it's very relaxing in the evenings. And for me, those creative projects are a really nice way to unwind and balance after a long day of work. And also, as most of the work I do is very technical, it's very nice to just relax and unwind a little bit.
Lovely. Yeah, I was actually just thinking that, I mean, you work with data, so it feels so natural it makes so much sense to have something to do that's more, well, shall we say analog?
Yeah.
Speaking of data, though, what does a data analyst actually do, especially in the context of bioinformatics and plant breeding?
Well, at the core, I mean, it's working with data, as the name says. Usually it's big tables or sequencing data. And the main goal is to find patterns, extract insights, and also answer specific research questions. In case of plant breeding, I identify, for example, if certain genes are present or what do they do, what is their function, analyzing which plants need to be crossed for higher yields, or identifying the best performing plants under certain conditions. So in short, it's turning raw biological data into knowledge that guides breeding decisions.
So can you give an example maybe of a specific project where you do that?
I work on different kinds of projects. I mean, I work on xSeedScore that you've heard about several times, I guess. There we receive genotypic and phenotypic data and also environmental data, so structured tables. I also work with metagenomics data, so there it's DNA sequence data, so raw text files with sequencing reads. The general workflow is always bringing the data into a useful format, also performing quality control steps, and then in the end, generating visualizations and reports to help the customers answer their personal research questions.
I think that could be especially interesting for our audience. What kind of questions are being asked? Or what can you give an example of a research question or a question one of your customer has without maybe naming names?
I mean, most of the time, the customers want to identify more varieties, for example, that are adapted to climate change. They need to know which plants they need to cross for more resilient or higher yielding crops. In case of metagenomics, a customer usually wants to see the kinds of bacteria that are in their soil, for example, see if they have plant growth promoting properties or if they're pathogenic, to get an overview of their fields.
So I think the platform you're using with metagenomics, mainly it's MORPHEUS, right?
Yes.
I love that name. Very Greek mythology. So if you mentioned as an example, people are trying to identify what's in their soil, how does that process work? So how are you working with the customers? For example, if that were the research question, what happens one after the other?
So it depends if a customer already comes to us with their raw sequencing data, or if they have samples, then we will try to find a sequencing provider for them. And then we receive in the end the data that we work with, which is the sequencing reads, the raw sequence data. And then a long process, a long bioinformatics process on our side starts that the customer doesn't have to interact with because there are several steps involved. We have our in-house pipeline to analyze the data, perform quality control steps. And on the way, we always have meetings with the customers also to inform them if something is maybe odd with their data. So they might send us more or a little bit higher quality data. But usually the workflow would just be that they send us the data and then we perform the quality control and analysis steps. in the end, reproduce data in a format that we can then actually create those reports. And my job is then to run those pipelines, also to maintain those pipelines, and then in the end create visualizations, dashboards in that case, that the customer then has access to, can play with, and then get insights into their personal microbiome that they want to analyze. Mm-hmm. So we then offer them just like this interactive way of looking at those reports, downloading the results in a way that is also useful for them and that answers their question. So it's just a huge dashboard of different types of panels where you can look at taxonomic content or functional content. So you actually know what is in your sample and also the function. So what does a specific or what does the microbiome do? Which kind of functions are encoded based on that metagenome? And also, for example, if there are pathogens in there, for most customers, it's also important to know like if they have pathogenic contents in their samples.
That makes total sense. You've mentioned a number of times that you do quality controls as part of this process, of this large bioinformatics process on your end. What does that look like? What does that mean? Maybe for someone like me who's not doesn't have a bioinformatics background.
So for metagenomics, it's a different workflow than for other analysis tasks, of course, because we don't have tables, we have those sequencing reads. We have our own analysis or quality control pipeline where we put in the data. I mean, this is a coded pipeline. So, of course, we maintain it, but for an analyst, we just put in the data and then we receive the output that we then can create also little plots with. So the customer, for example, then sees the read length of their samples, or if there are just like very low abundant samples, if we just have a very small read count in some samples, because in metagenomics also this can vary depending on the environment that you work with. Also, maybe during sequencing, something might have gone wrong. And then we have to decide, okay, should we continue with these samples, or is it possible to redo the analysis on their side or on the side of the sequencing provider?
So even in the quality control, there is a back and forth with the customer if needed, correct?
Yes, that's very important because the whole pipeline has so many different steps. And when in the end we see that something looks odd, it's always important to realize also right away in the beginning if some issues might occur. I mean, biological data is very complex and sometimes also quite messy. So it's important to also have control over all of the several steps to also identify where a bias, for example, might come from. And from time to time, like you will get experience and then understand also like what types of issues can occur and how they can be solved.
I mean, this might be a tough question, but can you recall like what was the most surprising? So this seems to be sometimes a little bit of detective work, right? You have to interview, you have to follow the clues. What could be the issue? Do you recall any such incident where you had to find out why are we getting this mistake, or why are we getting this weird data? Can you recall?
Yeah, I mean, we did have a case once where we thought you would get data of a certain type of sequencing technology, and our pipelines wouldn't give us the results that you would expect, and at some part it was failing, and we had then discussions with the customer back and forth, and at some point we realized that they have just used a different protocol that is not suitable for this kind of sequencing technology. Because we were also just wondering, okay, what has happened? Maybe something has gone wrong during sequencing, but it was just like wrong primers, like the wrong method just at the beginning.
Okay, so could this be comparable? So I'm trying to think of maybe people out there who don't have the background. Would this be like, it's still a video, but it has a different codec? So kind of the underlying protocol structure is just different and can't be read in that case.
Yeah, I think that might be a good comparison. So it was not the reads that we were working with just didn't have the length that we were expecting, and they should overlap at some point, and they were not overlapping because it was a different method that is not generally used in that way, and that's no problem. It's just sometimes difficult to find that issue, right? So to also identify why it's not overlapping, because usually we didn't get data that way, and it was the first time. But now we also know that we have to check for problems like this right away, just to prevent a lot of detective work, as you said.
For sure. It's interesting, though, because I think a lot of people maybe think because you work with AI so much, and as you said, a lot of them are automated pipelines, but it still takes a lot of humans with a lot of knowledge and a lot of experience to really make the pipeline work, right? And also a very close relationship to the customer. So I think this really shone through in the example you just gave us. You mentioned that outside of the metagenomics projects, like with Morpheus or other ones, you work in quality control is also very important, or you have maybe also a more general understanding of quality control. What did you mean by that?
I mean, when you work with tables and huge amounts of data, then there's a lot of missing values involved sometimes. Always when humans are involved, it means that there can be some issues, like naming issues. And when you have like thousands or even millions of rows or columns, it's very difficult to find that issues right away. You could read through it, but it would take super long. So we just have to do like internal steps to check, for example, for naming, if there are duplicates in the data, because that also causes problems. Yeah, I think mostly it's the naming, because if people just like write the data by hand at some point, of course, because they take notes, those issues can occur. But our customers usually also have a very good system internally, so we don't have to do that much detective work, but issues can always happen. And of course, then if we don't find them in the beginning, they could lead to weird outcomes. And usually it's just very simple issues then in the beginning. But also, like, I think the most interesting thing is always when you receive the data and you're just like a data analyst, you don't know the biological processes behind. Sometimes this might be very difficult. It's then always nice to also like have people with different kinds of biological backgrounds, because they might have actually done those experiments in the past. So you can also ask them like, okay, what could happen when you are doing a certain type of experiment or generate data in some way? Because when you actually create the data yourself, then you also know where the biases and issues might come from.
So that's the information you would also get from your relationship with the customers or from within the Computomics team?
I would say both. So, I mean, we have people here at the company of a variety of different backgrounds and also experiences, because, I mean, they might have done a project that I have not worked on before, but they have several times, and then I could ask them. But of course, also when we work with plant breeders, they do know a lot about like data generation and statistics and so on. So it's always also worth to ask them, like, how did you do your data generation? Because there might be differences between maybe different people, but it's both. So just having someone to know who you can ask is always good. And this usually works out.
And how does your work contribute to real world outcomes in science, but also in agriculture? Because like you just mentioned, plant breeders, they work with you to achieve certain outcomes.
Yeah, I mean, in terms of climate change, it's very important to find solutions in that way, right? So we do need fast data-driven methods to select plants that are giving us a higher yield, for example, or that are resistant to certain diseases. The whole field is evolving very fast. So also these methods are becoming part of everyday breeding decisions. But as I said, climate change is a real problem, and it's good to always also be part of that research and also be part of that work in the end to try to make an impact. As I said, find those varieties for the customers or help them better make decisions in that way to fasten up the process.
Did you already know, or was that part of the reason why you got into bioinformatics, or what's your academic background? How did you get into this?
I didn't start with bioinformatics when I started studying. So I did a bachelor's in biomedical sciences. This is then more lab focused, a lot of chemistry and biology labs. I really like that, but I also realized like working in a lab is not something that I want to do for the rest of my life. And I enjoyed the data analysis steps that are also involved in that area more actually than standing in the lab and producing those results. But yeah, I mean, I did come from more like a biology background that is more human involved. So going into the world of plant breeding or metagenomics with lot of bacteria is a different area. But when I studied bioinformatics, because as I said, I wanted to also learn more about handling the data, learning programming and so on, and had the opportunity to be an intern here at the company, I started to also fall in love with these kinds of areas because, I mean, it's also very, very interesting. I mean, biological data is always just really cool because it has this real life impact, and there's so many different fields also that you could explore there.
So then you made the switch to data analysis and to this field. I'm wondering now, with the job you do at the moment, do you do a lot of programming? Because you said you started in biology, human related. Now you're in the data analysis side, and it's obviously bioinformatics. So yeah, how much programming do you do, and how did you pick up those skills?
I mean, yeah, programming is part of my everyday work. I did learn the basics during my master's studies, but it's an ongoing process. So I learn new things every day also in that field. There are so many new methods. As I said, also with AI, it's so present. So we, of course, learn a lot of new stuff also in the job. And yeah, so I mean, programming is very important. I mean, I do not do hardcore software development, but also for those data analysis tasks, it's important to know how to handle the data. And of course, then programming is involved. Also, while like biology and what I studied there was more about memorizing and understanding facts, the bioinformatics or informatics part is more about problem solving. So you have a problem, you need to find a solution. There are usually a lot of different ways to solve this problem in the end. So this also is then very, very interesting. Sometimes also maybe a little bit frustrating when you stuck with a problem, but in the end, when you solve it, it's a very good feeling. So yeah.
I bet. And I know, or I happen to know, because of my inside info, that you also deal with agile project management or help to incorporate customers' wishes, because that can also be, maybe let's not call it a problem, but a challenge, right? If you have your system, but then maybe a customer comes along with a certain request. So how do you deal with that? Can you give us an example, maybe a practical example of how you attack that challenge?
Yeah, I mean, for xSeedScore, we have our own databases. And for that, we develop interactive dashboards also for our customers that are tailored to them. Which means that, of course, a lot of interactions with the customers and also internal meetings are needed. In this kind of project, we work together as a team. Some projects I just work alone and do the analysis steps. And then, of course, when it comes to customer communication, we are there also as a team from sales and so on. But in this kind of projects with the more agile way of creating dashboards and the database and so on, we have our small internal team. I really enjoy working in that area because it's always also nice to collect ideas with other people. But of course, in those weekly meetings with the customers, we always then present them the newest version of visualizations, for example. We get their feedback, and that also means sometimes that you have to redo something because a new feature has to be implemented that is not in there yet. But I also like that way of that it's not static. So it's this agile way.
It's agile and interactive and almost constant change, right? What would be such a feature that you had to change or that you had to implement in the first place? Can you give a concrete example?
In the beginning of the project, we had a nice overview of what we want to implement, but the way how to do that in the end, what the customer needs, is then something that becomes more clear in those meetings. Yeah, usually it's we, for example, present the customers a way to select certain plans or parental combinations, but then they tell us, okay, but we would also want the option to change the traits that are involved for a certain range, for example. And then you know, okay, I know how to do that. I don't know how long it takes, but then you just say, okay, we see what we can do. And then in the next or week after, we present them with another solution for that issue, and then they test it, and then they come back to us with another maybe improvement, or they're fine with it, and then we can continue to the next one. So usually it's more about like a customer wants to select something in a certain way, or they might also have their internal processes already, how they do selections. And then we want to mirror these selections that they already do internally. So they have a very easy platform to do that with all the steps that are involved.
That makes total sense. Maybe for a final question, from all you've told us, we can already tell that your job is very diverse. You have a lot of different types of tasks that you do and challenges you face. What part of your job do you enjoy the most?
I would personally say like the freedom to be creative in those projects, especially in those kinds of visualizations, for example. Also managing smaller projects is just nice. I really have fun doing that, and the wide variety of topics. We are a smaller company, so that also means that new ideas are always heard, which is nice. So you also know that you can make an impact, and that's something that I really enjoy.
Actually, that makes me think of another one. So if you could speak to people like you, or maybe your past self, would you have any advice you'd like to give your past self or other people like you who are maybe thinking of going into this field or don't know anything about the field? What advice would you give?
For someone who does not know how to start but is maybe interested in that field, if you're interested, then just try it out. Because what can go wrong? There's so many great resources online, so many free courses also to just try away from your bedroom. If you then realize that you actually like some data analysis and all these kind of things, then there are many, many great ways to learn and to evolve in that area. It's always important to be patient with yourself. In the beginning, I was also, I felt like I started too late with all the programming stuff because I didn't come from I didn't have a bachelor's in bioinformatics or anything, but also in this area, I realized soon that there are so many people, also colleagues, who also entered from different backgrounds. So when you really want to start in that area, the learning curve is quite steep, but it's always continuous. So you're never done learning, and that's fine. So don't need to compare yourself to anyone. Just take your time, and most important thing is to enjoy what you're doing.
I couldn't have found more beautiful words. Michelle, thank you so much for your time, for giving us an insight into your work as a bioinformatics analyst at Computomics. And we hope to have you back sometime, maybe to give us some more of that insight. Thank you.
I would love that. Thank you.
Thank you for listening in today. Want to learn more? Then check out our show notes and more info on Computomics.com. If you have questions or want to propose a next guest, please reach out to us at podcast@computomics.com.